In this video, we will discuss about how this Dasatinib blocks TGF-β induced EMT genes, or whether it is able to block them or not. Block or inhibit, we can say that. So first, in the figure on the left hand side, we will discuss whether this Dasatinib blocks the transcriptions of this TGF-β or not. So for this they used two different assays. First one is this P-Kaga assay, which gives the luciferase signals, and second one is this T-Plux assay. And what they did is they used dose dependent assay of both of this, that is P-Kaga and 3T-Plux. And what they are able to find out that with increase in the concentration of dasatinib, the relative luciferase signals or relative luciferase activity goes down. That can be seen here in the TGF-β1 impact. And at around 10 micromolar concentrations, the effect is almost negligible, and it is comparable to this inhibitor, which is SB431542. But when they did is using when they try to check this TGF-β inhibitions using bosutinib, they are not able to see any inhibitions of TGF-β, indicating that dasatinib works well compared to the bosutinib in terms of the reductions in the TGF-β transcriptions. Next, they wanted to check whether the dasatinib also impact the transcriptions of these EMT genes that are triggered by TGF-β. So for this, they decided to check some handfuls number of EMT regulated genes such as N-catherine, bimentine, MMP2, MMP9, slug, snail, and E-catherines. And what they are able to find out is that when they use dasatinib, they are able to see statistically significant suppressions of these EMT regulated genes. For example, N-catherines goes down both in the Colo357 and the Panc1 cells, and bimentines also goes down, and this decrease is statistically significant. This MMP2 also goes down in both the cases. MMP9 also goes down. Sorry about this. Yeah, and this slug is also goes down, though it looks like it's not significant, but they have shown here as the difference is statistically significant. And for this Panc1 cells, the difference is also statistically significant. And the snail's signals also goes down in both the cases, and E-catherine signals results are shown here for the Panc1 cells, and it shows that it goes up. And as E-catherines is the markers for the epithelial cells, it goes up in the case of these Panc1 cells when dasatinib is used in the TGF-β induced cells. And some of these results are also validated using the immunoblot in Colo357 and Panc1 cells, where they checked for the bimentines, that is N-catherines markers, and bimentine signals does not increase, indicating that Mesomical genes are not upregulated. And similarly, the N-catherine signals does not increase in the Panc1 cells, where we can see that it is not upregulated. And they also checked it for the snail signals. Snail signals also doesn't increase when this dasatinib is used in the TGF-β induced cells, and for both the Col357 and the Panc1 cells. Thereafter, what they did is they wanted to check whether this decrease is because of the AKT signals inhibitors, and this SB431542 inhibits the AKT signaling pathways, and they are able to see that significant decrease in the when they use this SB431542, indicating that this suppressions is because of the inhibitions of the AKT signaling pathways. And they checked for this slug pathway in the Colo357 and bimentine in the Colo357, and also the similar pathways results are shows similar genes results are shows for the Panc1 cells, and here also they see the significant difference. But when they check for this bosutinib, they do not see a suppressions. that is statistically suppressions or upregulations that is statistically significant. We can see that for the Colo357 slug and Colo357 vimentin. Similarly, the Pancon results are also look similar to the control for the Pancon slug, and these Pancon vimentin's results are also not statistically significant, indicating that it's the AKT pathway inhibitions that occurs by dasatinib, which is validated by suppressing by inhibiting the AKT pathway using these molecules, that is SB, using these inhibitors molecule, that is SB431542. So I hope you understood how they validate that dasatinib blocks TGF-beta transcriptions, inhibits TGF-beta transcriptions, and also the TGF-beta induced EMT genes, transcriptions of TGF-beta induced EMC genes. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.